Good morning, all. My name is Dr. Pete. I am a PhD biochemist, a nutrition network coach practitioner, and also a primal health coach. And this morning, we are going to talk about sugar addiction and the biochemical and drivers of this brain disease and its relationship to our organs and how the organs, from the taste sensors to the organs to the brain food reward system. We're going to talk about the variety of science that's out there that describes sugar addiction. And before we get started, I want to give you a little bit more details about myself. I'm not alone in this endeavor. I run a company along with my daughter, Tanner, who can't be with us this morning, but here's a picture of her here. And we are a team to help people reverse sugar addiction, along with other chronic diseases like type 2 diabetes, cardiovascular disease. I also specialize in the area of gout. In the show notes, I have put our contact information, so you can find our website at www.drpeteandT, all one word, .com. I also provide email addresses there. We run a program called Power of the Dog, and Power of the Dog is um, uh, connects to the concept of the red dog and the blue dog. And Power of the Dog necessarily refers to empowering the blue dog, developing our knowledge, which then helps us make decisions about our support. And in today's topic, in other forums, and even here, I've been asked, why do we need to even be concerned about the science? Because most of us know or understand that we have an addiction, right? We know that we're driven to eat the way that we're eating. So do we really need anything else in order to go down the road and get into recovery? And I think understanding the science is really, really important because, for one, there's more science out there than we realize from a lot of different disciplines. And as a biochemist, I was not trained to function in a defined hallway. When I look at a problem and I see our addiction to substances like sugar and alcohol, I see that as a problem. And if we want to solve that problem, in order to find out as much information about that issue as possible, this means that we need to look at as many different disciplines of science as we can to try and piece together a story about the biological drivers that make it as it seems so impossible to stop eating certain types of foods. And so that's really the purpose of my presentation today, before we begin it, is to understand two things. One, to understand better what's happening in ourselves because of these psychoactive foods. And then secondly, by understanding more about the connections between the biochemistry and the various organs that are processing these foods and substances, and then the brain reward system, and how we end up in an addictive state, is important for us so that we can make better decisions about our recovery. All right, so let me just check my notes here and make sure I've mentioned everything I need about the opening. Oh, and in the Power of Dog programming, we break this down into knowledge. That has a lot to do with my job, offering seminars like this one on what addiction looks like. And then later on in the coming weeks, at least once a week, we will offer a live video that deals with some aspect of recovery, some issue associated with recovery. And we'll talk about what we think the best practices are, which pretty much are going to match what you're hearing from all the other practitioners here. But we're going to break down the how and the why, and then the what in particular. We also have opened a free private support group on Discord, and we will be announcing soon when we will have peer collective gatherings there that are free. And they will be scheduled at a time where we can maximize not only the people who want access from the US, but also from Europe. So we're going to try to pick some times when the majority of people that are in this group can actually access this support group for free. And I would encourage you to go to that link and join today so that you're set. We will announce the schedule sometime later this week. We'll probably start with one group first, and then we will add others down the line as we get a feel for what the need is going to be. And feel free in our Discord support group to reach out to Tanner or I, both of us, Tanner and me, because there is an instant chat function to that support group. All right, so with that, let's go ahead and get started. So I'm giving a presentation on this today, which happens to be a poster presentation that I will be giving at Low Carb USA San Diego later this week. And I'm releasing the oral version of that early, exclusively in Sugar Bomb. Now, what I want to tell you guys about this today is that I want to be sensitive to confidentiality. So I want, if you have questions, feel free to ask them, but wait until the presentation is done and I will prompt you at that point. This way, when I download the recorded version of this, it will make it easier for me to edit off the questions, because the recorded version of this will appear on LinkedIn and on a YouTube channel that will be specific to Power of the Dog, which is coming later, like in the next week or two. All right. So with that, let's talk about now what I think is driving sugar and food addiction. And specifically, this is, I believe, fructose metabolism. And that's what we're going to talk about today, and how this biochemical driver is connected to the food rewards system, and everything that I can find right now that teaches us how these connections are made, and how the addictive process is actually being driven. So first off, my hypothesis is the following. When we think about addiction as a whole, and when you go out into the literature, you cannot avoid seeing all of the papers and the decades of data that exists for substances of abuse like alcohol, cocaine, heroin, and so on down the line. But when you step back and you think about, okay, sugar addiction, this is a complicated question, because sugar is not a sole substance, right? Sugar is composed of two kinds of molecules, glucose and fructose. And glucose and fructose are treated differently biochemically. So the point is that when you take and you eat a substance, or you eat a food that has sugar in it, the sugar itself can't be treated as being a singular entity. And when we think about where the biochemical driver will have come from, most likely this is a biochemical driver that has to do with the processing of food, right? And it's something that humans would have evolved very early on in the evolution of our species, because we have to eat. And when you think about eating, that in itself is not a generic thing, right? There's various macronutrients or molecules, for example, that are required for life, right? We need glucose. Humans can produce glucose on our own. So we actually, in terms of glucose, we actually don't need to eat it from the environment. This raises the issue about fructose and why it was so important in our evolution. And to understand that, you have to spend some time thinking about the difference in how glucose and fructose are handled in our body because they have different functions. Glucose is a compound that our bodies use to produce energy. And the energy that is being produced by the glucose, among other things, this central molecule called ATP, adenosine triphosphate, ATP. And you can think of ATP. I know we're talking biochemistry. Don't be intimidated. When we talk about ATP, we're talking about, you can think about it as molecular money, right? We go to the store in order to buy something there, like vegetables, something along those lines. We have to pay out money to get the vegetable, like a zucchini. All right. We can't just take it and run out of the store. We have to pay for it, right? And biological organisms, everything that we do as humans, the thinking, the running, whatever it is over the course of the day, we have to pay biochemical money for that, molecular money for that. We pay by using ATP. And the function of glucose, when it is coming into a human, right, we're eating, as long as the glucose that's coming in is coming in at moderate levels, then the glucose that's coming in is being used to generate molecular money, ATP. And that pathway has checks and balances so that when ATP levels are high, then the glucose is being funneled into that programming is funneled in more slowly. In other words, there's controls on that pathway. And we can argue overall that the reason that we need glucose to live is for a collective reason called burning. We need to be able to burn the glucose to produce the money, and then we burn the money in order to do the things that we're doing. So collectively, the glucose has a function in humans that I'm going to call burning. And burning is good for us. All right. Remember that burning is good for us. Now, what is going on with fructose? Well, fructose has a different purpose in humans. The input of fructose, its overall purpose is to flip the programming into a storage mode. So when the fructose comes into a particular cell, and predominantly we'll focus on the liver for a moment, as that fructose comes in, a number of things happen, and I'm going to talk about this more as we go through this. The first thing is that in the liver, as the fructose is inputted, we see a dramatic decrease in molecular money. The ATP budget of the liver is decimated, along with the phosphate that is needed to make more ATP. And as a result of the ATP depletion, we have a molecule called uric acid, which is then produced at a substantial level. And uric acid is a signaling molecule that drives the storage aspect or the effect that fructose has on the systems where it's being metabolized. So rather than making molecular money, the fructose, in a way of thinking, takes the molecular money away. And all of the inputting calories that are coming in at that point instead are pushed into storage. The fat synthesis with a decrease in fat burning, right? So we have this inverse relationship. We have fat synthesis and storage of fat. This is why we're going to gain weight. And we have a decrease in the ability of the liver to burn fat. So we have these two things going on simultaneously. In addition to our glycogen, right, where the glucose is coming in now, instead of burning, it's being stored away. So the body, when we have incoming fructose, has switched from a burning mode into a storage mode. And along with that, we're going to see there are going to be a cassette or a constellation of behavioral issues that happen uh, that are driven by the actual metabolism of the fructose, the signaling, which we're going to get to specifically, that is connected to the food reward system, and then modifications of the brain food reward system to favor increased intake of food, increased levels of hunger, the compulsive nature of going back and not being able to stop eating, and the behavior of going out and getting more food, which we call a foraging response. So what is my hypothesis? My hypothesis is the following. Number one, we need a driver. We need a biochemical driver because we're not injecting sugar, right? This is not the same thing as like a heroin addiction. We are putting a complex set of food substances in our mouth that have to be broken down and processed. And in evolution, we developed the capability to favor certain types of food macromolecules, the glucose, the fructose, fats, and amino acids. Today, we're going to focus on the sweets. We need a concerted signaling pathway. We need connections between the biochemical pathway itself and the brain, right? This information that's coming in as we're eating this material, or I should call it food, I guess. As we're eating this food, we need a way for the organs that are receiving this information to signal aspects of this to the brain. And then we have the food reward system there that needs to be responsive to these signals that are coming in. And as we dive into this, I just want to throw out one other thing to keep in the back of your mind. From an evolutionary point of view, as hunter-gatherers, our eating was more intermittent. So the biochemical pathway with its concerted signaling that goes to the food reward system was designed to be operating intermittently. It was not designed for the kinds of pressure that the food reward system is under now, where we have the standard American diet, right, where people are eating 5 to 6 meals a day, which is very, very high in added sugars. And in terms of the fructose coming in with table sugar, high fructose corn syrups, honey, and the processed food, which is always going to have added sugars in it, along with salt, and also along with a type of substance called umami, which I will get to. So, with the biochemical driver and then concerted signaling into the food reward system, a food reward system that is manipulatable, that can undergo modification. This is how we end up in the addictive state for certain types of foods that have certain proportions of these macronutrients. And today we're going to be focused on fructose. So as we get into this, the first thing that I want to talk about is that fructose metabolism has fairly grave consequences for humans, because we have a biochemical pathway that, as it's opened or activated in a human, we see this decimation of molecular money. At the same time, we see this significant rise in intracellular uric acid with its signaling, which is driving a host of chronic diseases. This is why people who are older, like me, I'm 64 now, who are suffering from sugar addiction, as an example, right, which is one of the consequences of this pathway, may also be suffering from other issues like gout, kidney disease, diabetes, Alzheimer's, fatty liver disease is super common. So the approach that we have to recovery, if we just step back from this right now, even before we get into the details of these connections, is in recovery, we favor approach that reduces the intake of the sugar, right? We reduce the intake of fructose. And by reducing the intake of fructose and also reducing hyperglycemia, not only can we recover from sugar addiction in the long run, right? And I'm not suggesting there's an off-on switch here. We have the whole recovery issue to talk about, the change in habits, all these things that take so much work, require integrating with other people and working together to develop new habits. I'm not saying anything negative about that. We need that aspect of this, but one of the issues that we need to do is we need to quiet this pathway down, right? We need to stop activating the food reward system, which is driving the hardwiring of the need to eat this certain way, right? We need to stop activating that pathway. And when we reduce the sugar and whatnot, one of the benefits that we get is the recovery from these other illnesses that are part of how this biochemical driver works. So first, let's talk about the biochemical driver, and we have to talk a little bit about the biochemistry of this. There's three main classes, of macronutrients that open up this pathway. One of them is eating fructose from table sugar, high fructose corn syrups, agave syrups, honey, and packaged food directly, because eating any of those substances contain large quantities of fructose, which I call exogenous fructose, because you're taking that fructose from a source outside you and you're eating it directly. And when fructose arrives at the liver, the majority of it will be moved into the liver with no feedback mechanism. And all of the fructose that comes in is going to be converted to fructose 1-phosphate. That point on its own is not as important as understanding that in order to convert the fructose into this thing that has a phosphate on it, you have to decimate the molecular money that is in the liver. So we see the substantial decline in ATP, which is one of the central drivers of the hunger that makes you eat more. Okay, and then as that ATP is plummeting, we have the significant rise in the uric acid on an intracellular basis inside the liver. And this is driving the hunger and the cravings, the overeating, the compulsive eating, the foraging response, which is driving you to get out of your seat and go to wherever that go-to food is and getting more of it. That's what's driving this. We also have two other contributing classes of substance. We have alcohol and we have hyperglycemia. And they activate this pathway, fructose metabolism, by entering this important biochemical pathway using what's called the polyol pathway. This is where, if we have alcohol and hyperglycemia in particular together, although hyperglycemia can do this on its own, above a certain threshold, can activate polyol. And what polyol does is it converts glucose to fructose directly in the liver. And this type of fructose we call endogenous fructose. It's being produced in the liver. And while we're talking about this, it's important to recognize that the metabolism that I'm talking about here is not just occurring in the liver. It happens, and this should concern all of us, directly in the brain. It happens in adipose tissue, skeletal tissue, in our intestines. We're going to talk about that more in a second, right? So we have the metabolism of fructose in the intestines, along with kidney, and then where we're talking about now in liver, and also in the joints between our bones, right? Knee joints and toe joints, finger joints, and so on, predominantly this metabolism most likely is also happening in the important biological cell there, which is responsible for the health of our joints, the chondrocyte. Okay, so in this slide, what I wanted to do here was outline together so you can just visualize this, that we have basically two kinds of things that are happening here. On the right in the red is the behavioral issues that are being activated when this pathway is activated. On the left, you can see what's happening on a biochemical level. So as that uric acid is coming up, we have a significant rise in systemic inflammation with the production of certain kinds of transcription factors like JNK1, which is a protein, a transcription factor that activates genes, that is sitting at the top of the inflammational cascade. We also have cytokines like IL-1 beta, which drives inflammation. And later in this discussion, we're going to be talking about what happens in the brain, where we have activation of a number of different proteins that drive the addictive state. In the liver, we also have an elevation of the disruption of the nitric oxide pathway, which downstream can lead to cardiovascular disease. We have an increase in blood pressure, and we have an elevation of insulin resistance, which means that our ability, as the fructose pathway is being activated, our ability to modulate the glucose, which is also in the bloodstream, becomes more difficult. So now let's turn to the signaling pathway. And this is super important because we can break this down into basically three areas. First is what's going on in our tongue. And we have three kinds of taste sensors that are important to driving the addictive state, all right, that are linked to driving the addictive state. The first is the sweet sensors. That's mainly what we're focused on today. We have umami sensors. These are specific for sort of those dark, rich flavorings. As think like eating an anchovy and how that tastes so rich and salty. Partly, we have the salt sensors too. Partly with the anchovies, it's because they're usually highly salted. But we also have individual salt sensors, and all three of these tastes are involved ultimately in the addictive state. There's a lot, what I feel is a lot of data about the sweet sensoring, and that's what we're going to be talking about today. But I think as time goes on, we're going to hear more and more about the umami and about the salt effect as well. So we have the taste buds with signaling to the brain. And the interesting thing now, that as we focus on sweet, and remember that fructose is significantly sweeter than glucose. This is one of the reasons why the food processors put so much of it into their food products, because it is significantly more active when it comes to activating our taste sensors. And taste is not just involved in preference. Like you put something in your mouth and you're like, that tastes so sweet, right? The taste buds are also involved in uptake. So the tendency to drive eating more of that substance than you should. And there is a relationship. There's enough data to say that there's a relationship between what's going on with the taste bud in terms of sweet and the liver function, which I'm going to get to next. So the liver is mainly functioning in the metabolism of the macronutrients that are coming in, the sugars, the fat, and the protein. And again, we're going to be focused now on the sweet aspect of this. So the fructose and the glucose that's arriving, and the metabolism that happens in the liver has been shown essentially unequivocally to have what probably is the largest role in the human body for driving chronic diseases, and is linked to the addictive state because the liver not only is connected directly to the food reward system itself through nerves, but it also produces endocrine factors. And one of those, okay, let me back up because I used sophisticated language. An endocrine factor is a protein that is produced by the liver that is released in the circulatory system. In this case, FGF21. This is fibroblast growth factor 21. I apologize for the name. I'm not the guy that did it, right? But this protein, when released to the circulatory system, makes its way to the brain and is involved in causing the satiety, right? The point at which you eat less, in particular of glucose. And I talked a few minutes ago about the taste sensors here. And when the sweet taste sensory system in the tongue is activated, there seems to be some data, and granted, this needs to be researched more definitely, but there seems to be some data that suggests that activation of the sweet by fructose causes the downplay or downregulation of FGF21, so that in the brain, the arrival of this endocrine factor is reduced, so that the signaling that we get by the brain, okay, you've had enough glucose, is not generated. And so this is one of the ways in which, in terms of the brain signaling, we continue to eat more. Now we move to the gut-brain axis, and this is really important because our intestinal lining has sweet taste sensors in it. And I want to get this right. So there's several categories that we have to pay attention to with the gut. The first is that it drives preference. So it prefers fructose and glucose, but its preference is higher for fructose. Remember that we also have in the intestines fructose metabolism, and this is driving in part the cravings that we have that come, during the meal while we're eating it. Wow, this is so good. I want more of this. So you keep going with it. But also over time, this is playing a part in the cravings that we have that are happening post meal. All right. And we're going to be talking more about that as we go here. And then we also have effects, and this is the thing that I want to emphasize here. We have an interrelationship between these taste functions, the taste buds in the tongue, what's going on with the liver simultaneously to what's going on in the gut, and also our adipose tissue. Because and there are different types of adipose tissue. I don't want to get into this right now. We're going to talk about this on a general basis, but adipose tissue can be considered an endocrine machine, and one of the crucial proteins that adipose tissue produce and release is called leptin. And when we activate the fructose pathway, the leptin is overproduced. We generate what's called leptin resistance. And what does that do? It causes an increased intake in food. So we don't stop eating. We don't get the signaling that we should get. We sit down to a meal, right? And we start to eat that meal, and we're not getting the signaling that says to us, okay, you've had enough and now you can stop. So there's a number of aspects to this that are all contributing to the function of overeating. And then we need to shift now to the brain in order to understand how the food reward system itself is potentially modified down the line with the production of more proteins like delta phos B, you can see in the upper right hand of my slide here, and CREB, cyclic AMP, element binding protein that are involved in pushing the addictive state. So let's move to the next slide. So first, let's look at an overview of the food reward system. And this is a very idealized slide. I've showed it before in some other talks that I've given. Um, it but I think it illustrates very well the relationship between the incoming signaling. This is the dopamine pathway. So as the signaling comes in response to the things that we're eating, dopamine is released. This is an adaptable system, meaning that when certain of these macronutrient inputs and their signaling happen, then there is a decrease in the dopamine response over time, which drives the person to eat more of that substance in order to generate the same dopamine response. And just to take you through this from an overview, if we look at the prefrontal cortex first, which is over there on the left, this is what, while we're eating, keeps us focused on that matter at hand, right? The material or the substances that we're putting in our mouth at the time. The nucleus accumbens, it is a brain center that is driving the locomotor, or literally the drive to the spooning function, if you will, to keep putting the food in your mouth. The amygdala, which is down and to the right, it is the organ that is going, I love this right now. Oh my God, this is so nice. This is so good. I just want to keep rolling with it. And then finally, we have the hippocampus, which is involved in remembering. And what does this have to do? It has to do with taking in all the sensory information around that food environment that we're enjoying so much, so we can find our way back to that environment so we can do it again. These are not the only brain centers that are involved in the addictive state, but I think this gives us an overview so that we can understand the next part of this. So this is a more, concerted diagram about the food reward system, the dopaminergic system, right? That is the dopamine that is being released in response to these incoming signals. And over time, what I want you to understand about getting to the addictive state, and this is the thing that sometimes we hear arguments about by the established medical community, by certain psychiatrists and whatnot, that want to tell us that, you are experiencing an addictive like state, but you're not really addicted, right? They start hand waving. And we understand that they're wrong. Okay. And their argument is based on the idea that the incoming, and I'm going to stick to the cutting edge of the science right now, that the incoming fructose is not generating in an individual meal the type of response that's required to cause the brain to undergo these adaptations that arrives at, the addictive state. And where they are incorrect is failing to realize that we evolved these pathways and they weren't designed to be operating on a chronic level. Now, when we ask the question, I'm going to be getting to this shortly, but I want to raise the issue now so that you can understand better what I'm saying. When you feed an animal fructose, proteins are released in the brain that have been shown by the addictive researchers, the guys who study cocaine, heroin, down the road we go. The same exact protein that drives the addictive state has been shown in many, many, many publications. There is solid agreement by that group of scientists that this protein, I'm going to be getting to it anyway, FOSB, delta FOSB, is driving the addictive state. It's also activated by fructose and by sucrose, which is a mixture of glucose and fructose. What's the difference? The difference is the delta FOSB is released to 40 to 60% of, in a sense, the activation level of taking in a hard drug like cocaine. Now, here's the thing that we need to remember about taking fructose in. The issue becomes the level of chronic consumption, right? That if you think back, I think if you sit back and you think about, all right, what would it be like to be a hunter-gatherer, right? Where your food supply is not a given. And so you come, I'm going to use a metaphor. You come to an apple tree, all right? This is a metaphor. It's not, I'm not making an argument that humans evolved on apples. No, I'm not doing that. Well, we come to an apple tree, right? And what do you think is going to happen? We would stop and we would eat our fill. Why? Because we were starving. It's really pretty simple. But that apple had a lot of fructose in it. So the fructose activates the pathway. Almost all those calories coming in as we're sitting there chowing down on apples will be shunted into fat storage along with water, right? Because water is stored with fat. Water is also stored with glycogen. So basically, as you start to eat these apples, we activate the pathway. All that stuff is going into fat storage, which allowed us to survive. Because when we moved from the apple tree away, we didn't know when the next time it was that we were going to be able to eat. And the reason why this allowed us to survive is that when we moved away from the apple tree, now we had fat stores that could be used. And as we burn that fat, and as we burn the glycogen that we gained by eating the apple, we also release water. And this helped us move from one food source to another, sometimes across regions that were low in water as well. There were lots of reasons why this pathway was so important to us. But now, fast forward to where we are now, and you got a guy who's seated, trigger warning coming up, in a fast food joint. And what are they eating, right? They're drinking something that has a lot of fructose in it, a ton of fructose in it. They're eating food that is processed food that is loaded with the other, all of these different activators, the salt, the umami, and more added fructose, right? And this is the way most people are eating now on the standard American diet. So this pathway is being activated chronically. And that means this protein, delta phosphate, is being activated and it's being kept high chronically, day in, day out, month after month. And there are other aspects of lifestyle which also activate this protein, and I'm going to get to it. And what is the function of phosphate? The function of phosphate is to drive that individual to do more of that behavior. So it's involved in driving the modifications of the food reward system to facilitate these behaviors that all of us out there in this group are working so hard to put behind us, right? And this phosphate is crucial and its activation is crucial to this transformation. What transformation am I talking about? The need for chronic signaling, which we have if people are eating this way. We have reinforcement of the preferred foods, the fructose and the glucose and the alcohol, because alcohol activates this pathway. And when you hear the practitioners talking in here about, okay, if you're in recovery from sugar, be careful about alcohol, which is really good advice. And why is that advice important? Because alcohol piggybacks on this pathway and can activate fructose metabolism, especially when you have the alcohol, the hyperglycemia, and the input of the fructose all at once over a short period of time. Like I'm talking an hour or two of uninterrupted drinking and eating. Kaboom! You have all three of those issues arriving at the liver, and the hyperglycemia and the alcohol arriving at the neurons that activate fructose metabolism. And bam, okay, we have the conditions to elevate this phosphate, which drives the the tendency of our food reward system to be modified and move towards the addictive state with the compulsive eating, the activation of transcription factors, the manipulation of the DNA in those cells, right? Because our DNA is not just naked DNA hanging out like railroad tracks, like a lot of us are taught in school, right? Waiting for a gene to be activated. That DNA is all complexed and wound up with proteins. And the way that it's wound up allows for the activation of some genes and not others. When we activate FOSB, what happens? Well, the way that DNA is packaged changes. Now certain genes are open that were not before. And these genes, this cascade, which is still actively being researched, is now active and contributes to driving us to the addictive state. This is super important. Number 6, neuron plasticity. All right. Sometimes it drives me crazy that with the way the scientists come up with these definitions, because you look at neuron plasticity and it's like, what the heck does that mean? Well, it means that in the food reward system, you have all these neurons that have interconnections. This is how the signaling happens. And now we have a driver for the addictive state. So neuron plasticity means that the food reward system is going to be manipulated. So there are going to be changes that happen. And these changes will happen over time, right? It's going to take time for this. But with the chronic introduction of these specific food sources, we start to maximize the connections for the addictive state, which is the reason why when we decide to do something about our recovery and we turn off fructose metabolism, we cannot expect recovery overnight. And for some of us, the time it takes to remodel those connections, right? Because that's what we're going to ask the brain to do. We need to deactivate this chosen pathway, and we need to foster new connections. And this takes time. And for some of us, we're talking years, right? Years of work to get these new habits because our circuitry was so driven into this setup, right? While others, it will take less time. That's a function of our genetics and the fact that we're all different individuals. So I've already talked a lot. The cat's out of the bag. Delta FOSB is super important. It's expressed in a lot of brain centers, but it's super expressed in the nucleus accumbens. And any of you guys that follow the food reward system story, being told from the other way, looking at the brain inward, right, with all the neuroimaging and things like that, understand that the nucleus accumbens is at the center. It's at the center of the food reward pathway. And this is one of the locations, maybe the central location, though, where delta FOSB is being overexpressed. And where has most of the research for this come from? It's come from the guys who are working on the drugs of abuse, right? The cocaine, the heroin, amphetamines, nicotine, alcohol, and so on. But recently, there have been studies, both at the level of animal and post-mortem human, that have shown that FOSB is activated and produced in the brains of these animals in response, and this is key, to fructose and to sucrose. Remember that sucrose is a combination of glucose and fructose. And while, in my opinion, it's still unknown what the relationship between glucose itself and fructose itself and FOSB remains to be seen. We know from at least one important paper that was published, whose reference is number 2 down here, that in particular fructose is capable of activating FOSB on its own. And the one thing that the drugs of abuse people all agree about, down to the last one of them, is that FOSB is driving the addictive state. And here we have it being expressed in when fructose is metabolized. You know, I mean, how much more data do you need, for God's sakes? There's enough here. And I'm not saying that this is a complete story. It's not by any means. And I'm super interested in what happens next with this story, right? But it's something that to me leaves no doubt about the relationship about what we're putting in our mouth and what the potential outcomes of that in terms of addictive behavior can be, and in particular to this evolutionarily important mechanism that involves fructose. Now, before I move to the next slide, I want to mention that we know that FOSB is activated by alcohol consumption as well. And don't forget, with alcohol, I'm only talking right now, and this is something I think that's new that's out there about the effects of alcohol that are different than what we know to be the brain effects of alcohol, right? Here we're talking about how alcohol does yet another thing, which is to activate fructose metabolism via the polyol pathway. Additionally, we see FOSB, and this is food for thought to you guys, food for thought. It's also activated by stress, people suffering from depression, and from exercise. And when I talk about the issue of exercise, I'm referring to animal studies in particular where they looked at wheel running, right? Wheel running. That's where they put a mouse or a rat on a wheel and the thing takes off, right? And so some of the skeptics out there are going to argue about that. They're going to go, well, we're not wheel runners. But again, when we talk about, and many of the practitioners here will talk about outlets, right? So if you have somebody that's addicted to sugar, right, there are usually outlets. And what are one of the outlets? Usually over-exercise. It's one of them. So again, this ties back to FOSB. Remember the evolutionary aspect of this, right? Humans were driven to certain types of food sources, and we were motivated to find those no matter what. So I don't see a conflict here between the fact that stress, depression, and exercise also activate this protein. I think if we have an open mind and we think about the constellation of the addictive behavior, and we think about these other aspects that are activating FOSB chronically, this whole picture really starts to make sense to me about addictive behavior. Okay, we have other transcription factors that are now known. These are other proteins that are activated on a level of DNA. The CREB specifically is involved in learning and stabilization of memory. Remember, on the food reward pathway, we talked about the hippocampus taking in all the sensory information so you can remember how to get back to that food environment, right? This baby right here most likely has something to do with that. We also have this thing called nuclear factor kappa B. Again, I apologize for the acronym in the name, but it wasn't me that did it. This is also an important protein that is produced, and one of the reasons that it is, if you think back to the beginning of my discussion where I talked about the fact that when you activate fructose metabolism, that you have a significant rise in systemic inflammation. This is one of the proteins that's involved in that cascade. And what do we see here? That there is preliminary data that involves this protein in the plasticity and the memory functions of addiction. What is the plasticity? It's the modulation of the food reward system, the recircuiting or the modulation of the circuiting to favor the addictive state. And this is one of the dudes that's involved in it. And when is it expressed? It's expressed during the inflammational cascade that happens when we activate this pathway. And remember, this pathway is not just being activated in the liver. It's being activated in the neurons directly in the brain. And that's one of the things that's really scary about FOSB and the proteins that we're talking about here. These are being activated in the brain. They're not being activated somewhere else. Well, actually, okay, I'm getting carried away because nuclear factor kappa B is activated in the liver during the inflammational cascade that happens, the systemic inflammation. But when we talk about the brain, what I mean to say is it's not just about the signaling that's activating the food reward system. It's also about the biochemistry that is actively going on in the neurons, right? With fructose metabolism there. There's also strong implications here for Alzheimer's disease. Now, I want to come back just briefly to this liver protein called FGF21, fibroblast growth factor 21. Sorry again about the naming. This is important because if this protein that's activated in the liver and then produced and released, so its target is the brain, if that activation and production is brought down, then the brain is going to lose the satiety signaling for glucose. Now, there's only preliminary data that this is happening, and it's a relationship between the taste signaling and the liver. And it's preliminary enough, I would have to say, that what I'm saying is an inference. But I think that it's important to mention right now that because this contributes to the satiety problem, so that when we activate fructose metabolism, that there are other drivers here too that are either being interrupted or that are being activated that are also in a constellation way of thinking that are contributing to the fact that when all of this is happening, that we don't stop eating, right? One last thing to mention before I close about FOSB is that it is a highly stable protein. If you get into the literature yourself and you start looking at how FOSB is produced, the gene that it comes from, what you're going to find out is that gene produces a lot of different kinds of FOS proteins. Delta FOSB is unique because sequences have been removed from it that would cause it, after being produced in the brain, to be destroyed after a certain time limit. The problem with delta FOSB, once it's produced, it's highly stable. So if you sit down and you have a high fructose meal, you're going to have the generation of FOSB, and it will be active. And this is where the time frame gets to be fairly gray area, but it will be active from a matter of 2 to 3 weeks to out to 2 or 3 months. So what does this mean to us that want to do recovery? Well, here's the thing. If you have one meal with fructose, you're going to elevate the FOSB. The FOSB is going to stay active there. It's driving the addictive state. So the cravings and all of the things that come along with that drive you to go back and have another one of those meals is going to be paramount there. But if we're eating on a chronic level, basically the FOSB is going to be at a fairly raised level for indefinite periods of time. And this is one of the things that we battle when we go into recovery, because the driving force to have another drink or to have that go-to food is going to be around for a while. And this is also the reason why somebody who's been in recovery for a long time, right? They have whatever happens in their life and they suddenly find themselves, in that situation where they're having their go-to food. And now it's like blammo, right? The drive to go back is strong and persists for a long time, most likely because of the activation of the FOSB. which seems its activatability increases the more that you do it. All right. And with that, I would like to thank everyone who's here, who's listening, and it's time for me to go. If you have any questions, feel free to email me with them. Like I said in the show notes, I want to make sure I maintain confidentiality here. And I see in the chat that it's only listing Facebook users without names. So that's good news. But what I'll do is pull this MP4 down. I'll remove off this latter talking that I'm doing right now that is not in the presentation, so nobody has to worry about being exposed to the outer world. So watch for our upcoming presentations. We're going to be doing them once a week. We have a support group. Feel free to join our Discord. It's free, and we'll announce scheduling to bring in as many of the U.S. and European visitors. We'll try to find a time frame in there that's going to work for everybody. Anyway, you guys have a great day. I'm going to go ahead and sign off. Thank you very much for your attention.